Welcome to Bench to Bedside, a weekly series of live conversations about recent advances in cancer, from the research bench to treatment at the patient's bedside. And now, your host and the director of the University of Kansas Cancer Center, Dr. Roy Jensen.
Welcome to Bench to Bedside. I'm Dr. Roy Jensen, director of the University of Kansas Cancer Center. My guest today is Dr. Andrew Godwin, Kayuu Cancer Center's Deputy Director and the Director of Molecular Oncology for the Medical Center. He is also a leader in the field of precision medicine, which is an approach to care that allows doctors to better tailor healthcare to patients' individual genetic differences. So, Dr. Godwin, thank you for joining us this morning. So what is precision medicine exactly?
Well, as you know, doctors have known that patients are individual and different, but they haven't had the tools to treat them as individuals. And so what precision medicine, sometimes known as personalized medicine or molecular medicine, is really the application of science and technology to basically advance our understanding of health and disease at the molecular level. So, as you know, what we've tried to do in the promise of precision medicine is really to give the right drug or treatment to the correct patient at the right time. And so, really, based on that, we're using molecular diagnostics as ways to advance our ability to understand patients at individual levels so that we can treat them more appropriately and have better outcomes.
Mm-hmm. So the concept of precision medicine has been around for a while, but is really beginning to receive more and more attention over the last few years. It was even mentioned in President Obama's 2015 State of the Union address. Why is there the increased interest now as opposed to, say, 5 years ago?
Well, yeah, as you realized and mentioned, precision medicine or translational medicine has been around for a long time. And I think the interest has really increased over the years, and especially because of big advancements. So, to date myself is, one of the biggest scientific advancements when I was growing up was man landing on the moon. And now, as a molecular biologist or a cellular biologist, I never imagined that we'd be able to sequence the genome. And in 2001, the first blueprint of the human genome, which is basically the 3 billion letters, repeated letters of TCGA in different order, were accomplished. But that took 13 years to do, but it gave us a blueprint of the human genome. After that, then, we started to do work, and I was part of the Cancer Genome Atlas called TCGA that sequenced human cancers, and we sequenced 33 different kinds of cancers and over 11,000 tumors. And by generating that blueprint now of cancer genomes and understanding the molecular changes, the mutations, the unique differences between patient samples and across different kinds of cancers, so that an individual cancer, breast cancer, is not the same between individuals. We started to understand that we had this new tool, meaning the cancer genome, that we can now look into and identify unique changes that we could use for therapeutic treatments. And I think that when that project was completed in 2017, again, it was about a 10-year project, we started to, even before that was done, we started to utilize that information. And I think that that's really what brought precision medicine and oncology forward, by having the ability to have that blueprint and being able to really recognize the fact that an individual tumor is unique, and that each person's tumor has different features to that, and that we can start to explore about what we can look for, the Achilles heel of a tumor, in order to treat it more specifically and basically advance from our common way that we treat everybody with the same regimen, where we treat everybody with a chemotherapy regimen. We can now potentially target specific alterations within that tumor and have better outcomes.
Mm-hmm. So maybe could you give us a specific example of how this has worked for our patients? And so basically, how has precision medicine changed treatment or diagnosis in some way?
Well, you know, the first really example of this that really spurred the whole field forward, and I was fortunate enough to be at the institutes where this took place, was the study of a leukemia, blood disease known as CML. And what was discovered in 1960 by David Hungerford and Peter Knoll. David Hungerford was at the Fox Chase Cancer Center, where you know I spent 26 years of my career. And then Peter Knoll was at the University of Pennsylvania, where I also studied. Discovered when looking at these leukemia patients that they had a alteration which they could look at under the microscope. In this case, because the DNA is packaged into chromosomes, they noticed that one of the chromosomes was shorter in these patients. And it took another 10 years to identify what that genomic alteration was. And that was a fusion between two genes, one called BCR and the other one ABL. And that fusion gene was really one of the first discoveries of something that would be considered a driver, meaning that this was the cause of this disease. And what they found is that 95% of these patients had that genetic alteration. Fast forward another 10 to 15 years, Brian Drucker at Oregon State or Science and Health Center did a drug screen to find a drug that actually could target that genomic alteration. We call it a fusion, where a piece of DNA from different parts of the chromosome come together to make a abnormal gene that leads to an abnormal protein. And what Dr. Drucker found was a drug known as Gleevec. And it was remarkable when Gleevec was first used into the clinic that they had a response rate that they had never seen before in this disease. And now patients that have a remission on 2 years on this drug are pretty much cancer-free. And that really was the first example from 1960, a discovery, all the way to FDA approval 2001 of Gleevec for that particular type of cancer. Now what we've learned is, though, that other cancers are much more complex, have many more different changes, and don't have a single driver. And that's what makes some of the discoveries we made early on with CML doesn't translate necessarily to all types of tumors, because the other types of tumors are more advanced in regards to genomic instability, the level of mutations that are present.
Mm-hmm. So I think one of the major lessons for me from the TCGA project was that a confirmation that really each patient's cancer is a unique entity that we've never seen before. So how are genetic changes in a patient's cancer identified, and how does this guide treatment decisions then?
Well, as I mentioned before, is initially the discovery by Dr. Knoll and Hungerford was through doing cytogenetics, and that's looking at the chromosomes and actually being able to see physically changes in the DNA where it's moved around. And still we use cytogenetics as a tool. There are many ways, and we now have additional techniques that we can do to look at large, and those are more global, gross, bigger kinds of changes. But what we do in our clinical lab, for example, is that we can do sequencing of genes, whether individual or numbers of genes at the same time. So we now have the capability, based on technology that's developed over the years, to do what's called next generation sequencing. And what that simply means is, instead of sequencing one gene at a time or one region of a gene at a time, we can sequence tens of hundreds, if not thousands, of genes at the same time. And now in the entire genome. But from a practical standpoint, sequencing the genome for patient care is a little advanced, because we don't understand all the changes that take place that we can use, and we don't have enough drugs to actually address a lot of these genetic changes. But what we have now are techniques that we can do very rapidly and turn this around, where we can sequence patient samples where we get either the normal tissue or tumor tissue, and we can do that sequencing within days, if not less than weeks.
Mm-hmm. So just as kind of a follow-up to that, Given our current state of knowledge, does precision medicine apply to every patient that we see?
Well, what's interesting is that it applies, I think, in every case when we're talking about the field of oncology. We're learning more and more every day that there are unique changes. And as we work with the pharmas, as they develop new drugs that target those specific ones, we can start to see that we can apply aspects of precision medicine to almost every type of oncology. We're involved in early studies now looking at rare cancers, which haven't been studied nearly as much as things like breast, ovarian, lung, the more common solid diseases, or some of the, like AML or CML or other hematologic malignancies. But what we're finding is that as we go deeper and deeper and understand and use those molecular tools to uncover kind of changes that we can look at, and again, we're talking right now in only looking at the DNA, but there's many other kinds of studies going on looking at proteins that are, again, coded for by genes that are very important in regards to being able to monitor potentially disease. We're now developing new technologies where we can track potentially disease burden, another type of precision medicine, by using the blood or other bodily fluids, which are commonly referred to as liquid biopsies. So as we go forward, most every kind of oncology patient could have some degree of testing, whether it's a partial sequencing of their genome, whether it's measuring protein levels in their serum. There's a variety of different kinds of tests. But what I see for going forward in precision medicine, though, that will span almost all the different kinds of disease that we study, will be something referred to as pharmacogenomics. And that's really understanding how drugs are metabolized and being able to do tests to understand that certain drugs are metabolized by some individuals more rapidly than others, and that will help us give better and more accurate dosing to patients. Mm-hmm. And so I think the idea of gene-drug interactions is going to span all disciplines and affect not only oncology patients, but most patients that are coming in that are receiving any kind of drug that might have a different way that it's metabolized within the circulation in the blood.
Mm-hmm. So if you're just joining us, we're talking about precision medicine, and Alicia Miller is here in the studio to take your questions. Remember to share this link with people you think might benefit from our discussion. Use the hashtag BenchToBedside. So how are we using precision medicine at the Kayuu Cancer Center?
Well, when I came in late 2010, one of my objectives here was really to set up a clinical laboratory that would support molecular or precision medicine at the clinic level. Really that we're treating patients, we're receiving patient samples, we're analyzing those patient samples, we're interpreting the results and then feeding those back to the patient's medical records so that the physician can better treat that patient with as much information that they possibly can have on hand. And so in 2012, we established what's referred to as the Clinical Molecular Oncology Lab, which is a CLIA, College of American Pathology accredited laboratory that does patient sample testing on a daily basis. We offer many different kinds of tests, mostly based on RNA or DNA, many of those based on, again, next generation sequencing technologies. We've sequenced or evaluated more than 12,000 patient samples since I've been here, and we've used those diagnostic tests to help with the physicians to be able to use that information to better treat the patients. We continually are asked to support clinical trial development here. And for example, just yesterday I was contacted by one of our oncologists asking, can we do this particular test? And my answer is yes, we can. And what that test will do is it's looking at a single gene, and the patient will be eligible if they have a mutation in that single gene for a new therapy that's going to be tested for the very first time in patients. And so this is something that's changed dramatically since I first got into translational medicine more than 30 years ago, which is the idea that now we're selecting patients based on their molecular profile to be enrolled now in some of the newest clinical trials to try drugs that, like Gleevec, are targeted to a specific alteration. So this is really something that we're seeing more and more now. Instead of what we've historically done is treat all the patients the same. So giving of a drug could be a very active, targeted drug, but only seeing a 10% response rate. The goal now is to identify those 10% of patients before we give them the drug, and then give the drug only to the patients that are going to have the greatest benefit.
Mm-hmm. So what excites you most about the future of precision medicine in oncology?
Well, you know, I think that, you know, we always try to get, you know, I try to tamper the enthusiasm about certain areas because, you know, we're making strides, but we're not there yet. We're continually trying to understand the genome. And what I mean by that is that, you know, if we're talking about a genome of 3 billion bases, and that only 1% of that codes for genes, and it codes for about 20 to 24,000 genes, and that only a fraction of those genes are involved in cancer. You know, there's still a lot that we don't understand about the genome that actually could influence how cancer arises that we just don't appreciate right now. And so what my enthusiasm is that there's a lot of effort now to try to understand more and more about doing functional genomics, understanding why there are differences between individuals and what those individuals, you know, what those individuals changes due to risk of developing disease and protection from developing a disease. And so my enthusiasm really is in the area that, you know, we're seeing that have fruition in the clinic now. We are making differences, but we still have a long way to go. As I indicated before, certain cancers that we treat have a single driver or a few drivers, meaning that we have drugs that match those drivers and we see very good clinical benefit. But in other cases, we still don't quite understand all the changes that are important, and we might not have drugs right now that can target each of those. So as we make discoveries, again, you know, we can then help translate those into new drug development so that we can have, again, a larger cadre of drugs that will be effective for a given patient or patient population. And that's why I said right now is that, you know, as we're doing testing, you know, we're focused on what we can do right now to influence it. And that's what, you know, we're treating patients with, is the drugs that match certain genetic alterations. But, you know, from a research standpoint, we still have a long way to go in understanding all the changes and how they work together and how we might be able to start to address those changes early in the disease course instead of waiting till it's a late stage of disease where it's spread throughout the body. And now they have many, many different genetic alterations that we just don't have the drugs to treat at that point.
Mm-hmm. Alicia, do we have any questions from our audience?
Yes, we do. The first question, do patients have to ask their doctors specifically for the type of testing?
Well, in general, what we see is that one of the advantages of being in an academic cancer center like the University of Kansas Cancer Center is that we have very knowledgeable physicians about what kinds of tests are available and when they should be applied. And so again, you know, not every test is applied the minute a patient walks in the door. And so a lot of that is dependent on the physician's expertise and understanding about how you can use that information. Just having information is not necessarily of value unless you can utilize it to improve the patient's care. So our physicians, again, are some of the leaders in the entire country in their types of cancers that they study, and they're very knowledgeable about when they should order it. And like I said, you know, I get emails and phone calls daily asking about, you know, can we do this? What kind of, you know, what's the turnaround time for a test that we can do? And, you know, a variety of back and forth with the physicians because they're very knowledgeable and they keep up and they're leading, you know, they're presenting at major meetings about some of the findings that they're making in regards to, you know, these clinical trials that we're running based on the molecular profiles. And they really are the ones that are making the decisions when that test should be ordered. And our job is to basically do the analysis and get it back into the hands of the physician as fast as possible so that they can act upon on the findings that we discover looking at the patient sample so that something can be done immediately in that regard.
Mm-hmm. Alicia, any additional questions?
Yes. How many cancer genes have been identified, and are there treatments to target all of these genes?
Well, that's a really good question because that's a moving target. Is that, as I indicated before, that we estimate still, and it's kind of interesting, even though we've sequenced the genome many, many times, we don't know exactly the number of genes that are present. As I indicated, that there are 24,000 genes in the genome. Not all of those, and just a very small fraction of those are involved in cancer. And that's what we continually understand.
Well, no, wait a minute. No, I was reading the respected medical journal, I think it was the playbill of the Kaufman Performing Arts Center, where they definitively said that there were 20,000 genes, cancer genes.
Well,
is that wrong? Are you telling me that that's wrong, Dr. Godwin?
That's absolutely wrong. But as you know, genes have influenced our color of our eyes and our hair, and why you're so tall and I'm devilishly handsome. And but what we know is that as we study more and more diseases, again, and we haven't studied all the types of cancers, is that we are uncovering changes, mutations in genes, but there's also regulation of genes, meaning that some of these genes aren't expressed. There's different ways that they control the expression. Some of them are because there are changes in the number of copies of that gene. So normally in a human genome, there are two copies, one from mom and one from dad. And in cancer, a lot of times you'll get multiple copies of those. You could have dozens in some cases. Like, for example, in breast cancer, we've known for many years, again, that the number of copies of HER2, a gene called HER2, is very predictive of response to a therapeutic drug called Herceptin. But what we continually learn is that only a fraction of the genome actually have genes that control pathways that contribute to the pathogenesis or the development of cancer. And so currently, most screenings that we do are up to maybe 500 or so genes, and I still think that that's in the outer limit because we don't have drugs for all those genes at this point. So a lot of the work that we do is to focus on only those kind of alterations that we have that could be prognostic, could be predictive, can be directly linked to a drug. And so ours are focused so that we're not doing experiments on the dime of the patient or the insurance company. Because recently I just talked to a company that is sequencing the whole genome and doing something called RNA-seq, and the bill is $34,000. Well, the information you gather from doing that is only a fraction of that can we act upon at this day. So I look at that as being research. So if you're actually billing patients or insurance companies to do research, I don't believe that's appropriate. So what our focus has always been on is testing those genes or those biomarkers that we can currently act upon, that we have something that either predicts likelihood of recurrence, that we can predict a drug, and that's where I think that's the focus. But clearly there are not 20,000 cancer genes, but there are probably at this point, people will argue anywhere from 200 to 400 kind of cancer, really relevant cancer genes, but what we tend to focus on are really the ones that are linked to therapies or outcome predictions, those sort of things.
So what do you think about the claim that genomic testing should be done on patients right now?
Well, I mean, I think that we're seeing some
full-fledged genome sequencing.
Oh, full-fledged genome sequencing, I think, is important from a research standpoint. And that's what we've done, and that's what the Cancer Genome Atlas project was done. And again, that started out as a $100 million pilot project that then invested another $275 million, I believe, to do the completion of that study as the cost of sequencing came down. But that's research still. As I indicated before, we don't understand all the nuances of the genome. There's a lot of information there that we just don't understand how it affects development, how it affects development of diseases, and those are areas that I've been passionate about for many years. I was what's called a gene cloner in those days, in the early days where we were I was involved in trying to identify the first hereditary breast cancer gene, BRCA1. But you know, that's research in that case. What we try to do is then take that research that has the best implications to patient care and then move that into the clinical setting. So that's why we always have to be very, you know, work very closely with work that we do and others within the, you know, within the world, the United States and the world, about new discoveries that can be translated into the clinic and that can actually benefit patients. Because again, payers only pay for certain kinds of tests that have enough clinical evidence that is improving patient outcomes. And sequencing the genome has not demonstrated that you can, that alone is enough to influence the patient outcome. So for example, as I indicated before, the clinical trial that I've been asked to help support here is looking at a single gene. I don't have to sequence the genome to find that mutation in patients that then would make them eligible for that trial. We can do more focused types of approaches at this point until we then find that, you know, again, there might be new discoveries, which they are happening all the time, and I'm always being informed by our oncologists that we're going to need this test because there have been, you know, something at a major meeting or a major paper that comes out that's going to improve how we can take care of our patients. And my job is to, you know, provide that information to them and to the patient so that they get better care. But at this point, you know, randomly sequencing, you know, the whole genome on patients or the whole exome, which is all the genes, encoded genes, is not being utilized to treat patients, because a lot of what you find there are the things that we can find by doing more focused gene evaluations.
Well, I'd like to get your comment. I was always taught in my training that ordering a test and billing the patient to fund your research was fundamentally unethical. Would you agree with that?
Oh, absolutely, and that's why we've tried to keep that very much separated. I just had a meeting with somebody from another institute, and they're testing their lung cancer patients up front, every single one that comes in. Well, that's not being reimbursed. So the institute itself is picking up the cost because they think it's important to have, you know, that upfront information. And so from an institutional standpoint, we have to appreciate that. And we've done the same thing within the Kayuu Cancer Center and the Kayuu Health System, where there are certain ones where physicians want a test done, but it's not going to be reimbursed. But that's now being covered internally. It's not being passed on to the patient. And so those are important separations we have to do, but those are things that the clinician want, but at that point they might not be reimbursed. So, you know, we're trying not to put that burden onto the patients. And, you know, because again, as you know, financial toxicity is a true issue with patients who are undergoing cancer therapy. And doing the molecular testing is something we don't want to add to that cost, because we know that many of the oncology drugs that we're talking about this day are extraordinarily expensive. So our goal is to always keep our costs down, and when reimbursed, we get reimbursed. But when it's not something that's reimbursed, a lot of that is covered by the cancer center or the health system.
Do we have any additional questions, Alicia?
We do. The first question, do patients request the molecular testing, or is this something their doctor requests? And how can you be sure that your tumor is tested?
Well, again, we had a similar type of question. Yes, the physician will make the decision when a test is to be ordered. That report then, once it's ordered, it will go through a system. It then will go through the Department of Pathology. That sample will be obtained, and the sample will be sent to our clinical lab. The testing will be done, and again, the turnaround time varies for certain tests. Certain tests we have to turn around within 48 hours, other ones within 7 to 10 days. That information is then put back into the patient's medical record, and we alert then the physician that that test result is available. At that point, they will then talk to the patient about the test results that were done that could then help, again, guide the kind of therapy that we're talking about. In certain cases, you know, patients will be in remission, And we do a test that basically every time that patient comes in, we're doing a blood-based test to see and validate that that patient's still in remission. Those will go back, and I will tell you, it's just like any patient that comes in, like an ovarian cancer patient wanting to know their CA-125 level. You know, they're worried about that because we're the ones that can inform the physician whether that disease is in remission or not. But that information is then directly shared back to the patient to kind of, again, guide therapy. They'll be coming in for their checkup, said, you're still in remission, you know, no issues, or if we see an early recurrence of the disease, they can then decide upon another course of treatment. But each time we're doing this is that, you know, that information goes right back into the patient's medical record, electronic medical records, and the physician is then utilizing that information to better share with the patient about treatment options.
Mm-hmm. Our next question, Alicia?
Yes, the next question. The clinical trial that was just mentioned previously, is that available to local patients?
Well, it would be if they would come. It's all going to be dependent on the type of cancer they have, and after we do the molecular profile, if they would have that specific alteration. So if they would come to our institute, and again, this will be one of the first in human studies. It will be a very specific kind of cancer. There'll be eligibility criteria, and one of the eligibility criteria would obviously be that they have the specific genetic alteration. And then that patient then can, if they're coming to our cancer center, be enrolled within that study. If they meet other eligibility criteria, as you're well aware of, there are many that go forward. But it's not something that will necessarily be open at community sites. It will only be open within our cancer center here. But those are the things we're doing now. And you know, again, we're going to be opening a study again where I'm going to be for pancreatic cancer, where I'll be doing a screening of about 100 patients to identify 18 who are eligible. And this is another example where the cancer center is paying for doing the upfront screening. We're not charging the patient for that screening. And then those patients who have that specific alteration then can be enrolled. So we're going to have to screen about 100 to find 18 patients who will have that specific molecular characterization that will allow them to be on the trial. But those are the sort of things that you can get at an academic cancer center here, where we're always working with pharma or individuals within our cancer center to develop their own investigator-initiated trials, where we have, you know, some unique opportunities to treat patients based now on their molecular profile up front. And this is not, you know, after they've metastasized or something like that. We're now doing that as an upfront requirement for enrollment.
So one final question. What do you think about the ethics of touting whole genome sequencing as a way which will help determine a patient's treatment protocol when in fact there's very little evidence in the medical literature that that's the case?
Well, it was quite interesting. I was just on a AMP webinar yesterday, and they were reviewing some of the approved criteria for identifying patients, the kind of testing you do, and this was a study on lung cancer, both non-small cell lung cancer and small cell lung cancer. And what are the tests that are approved? And it was extraordinarily small. You know, there's only a few genes that would be tested, you know, less than 12. Mm-hmm. The way they're tested is a variety of different methods. But here's an example where this is the only thing that experts in the field have approved. It's not looking at 20,000 or 24,000 genes. It's not looking at, you know, 3 billion bases. It's looking at the genes that we know that we can influence, if they have those alterations, that we can make clinical decisions on. And so I think that you know, again, there's great value in doing the whole genome approaches or whole exome, and that's again sequencing all the genes, but as a research basis. And I've been involved in those kind of studies, again, funded through the NCI and the NIH. And many institutes are funding their own kinds of operation for discovery there. But from a clinical perspective, you know, if you're doing those tests, you're only using a small fraction of the information you're gaining. In the future, you might be able to go back and mine some of that information out as the patient might advance or those sort of things and see different changes. But at this point, it doesn't have any implication about how we make treatment decisions because they just, we don't know enough, and that's why that's the honesty. And so, I don't over-tout precision medicine because we have a long way to go. But there are certain tests that we can do, and as you know, I did some of the very first studies of a drug that targeted a protein referred to as the EGF receptor. And what that simply was is we identified that if we had mutations downstream, so this is a signaling pathway, so we have mutations downstream, that if you had a mutation downstream, that the drug upstream would not work. That allowed us to not give drugs to patients who wouldn't have benefit. But again, it's looking at a small number, focused type of one, knowing what the pathway ends up being. Studying 24,000 genes would not have helped us in that particular case because it would not have led us to understand that specific pathway any better because many of those changes would have just been noise.
Well, thank you, Dr. Godwin, and that is all for today. Thanks for that great discussion. And please tune in next Wednesday at 10 a.m. for our next episode of Bench to Bedside, brought to you live from the Kayuu Cancer Center, where we sleep well at night because we do what's best for our patients every single time.